good to go.
Okay. Good afternoon, everyone. It's my great pleasure to have Professor Andy Benson here with us to give us a talk on genomic analysis on human gut microbiome. My great pleasure to introduce Andy. Professor Benson received a PhD in microbiology from University of Texas Health Science Center. And then he conducted as an NIH postdoctoral fellow in molecular biology at Princeton University. And he joined the University of Nebraska as an assistant professor in 1996. And currently he's a professor in the Department of Food Science and Technology. In 2010, he was named as W. Marshall Distinguished Professor of Biotechnology at the University of Nebraska. And then in 2017, he was awarded as an Allen Food for Health Presidential Chair. What is most impressive is he is a fellow of the American Academy of Microbiology, which he was one of the only few that I've known as a scientist that was elected as a fellow in this academy, and who are working in food science. Dr. Benson's research has been focused on application of using population genomics. Earlier, his research was focusing on enteric pathogens. And I remembered in 2005, I went to the American Society for Microbiology general meeting in Washington, DC. And that was the first time and I met Andy in person, and I was in the audience, and he was presenting his research on genomic analysis of pathogens. And I was so impressed. I didn't know that genomics and bioinformatics can be used to study pathogens. So he is a pioneer in this area, and one of the very few food microbiologists in the nation and in the world that I truly admire. Since I was a student, since 2009, his research started to switch into more microbiome type of study with a focus on gut microbiome. And he established the Nebraska Food for Health Center and was has been the director since 2017. So without further ado, I would like to have Andy to take over and give us a seminar. I invited faculty, staff, some FDA scientists working here at IIT and IFISH. Also, I invited some good colleague of mine from different places. John Hong is from University of Maryland, probably there will be a few others to join from Cornell, University of Georgia, University of British Columbia, and so on. So don't be surprised. Okay, thanks, Andy.
Wei, thank you so much for that very, very kind, generous introduction. And it's been a while. It's good to see people that I know the names and I know the faces, but after moving on to a different field, you don't move in the same circles anymore. And so you don't get to see them very often. So it is good to see old friends. So I really appreciate that. As Wei said, I started off using population genomics to study foodborne pathogens and spent several years, the first part of my career doing that. And it was a lot of fun, I have to say. Understand the ecology and evolution of these organisms from a genomics perspective, and using population genetics as sort of the approach to combine with genomics. One of the things we ran into doing that kind of research was, and I'm sure some of you will appreciate this comment, is that we were in a position where everybody loved us, but nobody wanted to give us money, because we were very fundamentally focused, which was difficult for USDA to swallow. And a lot of the organisms we worked on didn't have good animal models, and so NIH didn't like it either. So it was always a challenge keeping some of that funded. And so for those of you that have stayed in the air in that area and maintained that trajectory, I applaud you for being able to do that, because it was always a challenge. But I do miss those days. They were fun. Well, I want to take you on sort of a journey of where I've been since then. I thought it'd be kind of fun to pick up and show you how and why I changed fields, and then show you a little bit about what the center's done and some of the work that we've published just within this last year. So I start with a timeline here. Around 2006, 2007, that's when next generation sequencing was commercialized. And the commercial products from those early days started to become available. So 454 sequencer produced by Roche, for example, the GS50 that was out there on the market also. Those technologies became available, and we realized in the context of a food science department, and this is a picture of Bob Hudkins and I right next to one another there, we realized that this technology would allow us to interrogate the gut ecosystem in a way that we've never been able to do before. Microbiologists have known for 200 years that was a complex community, but never had a good way to study it, and never really even understood how complex it was until we had an analytical method to study it. And we recognized that this next generation sequencing technology was going to be that analytical tool. And so we started something called the UNL Gut Function Initiative. We wrote white papers to our administration. We had a couple of new faculty hires that the university invested in. That's Jens Walter right there in the middle. Jens was with us for 10 years. He's a microbial ecologist. He's since moved on now, and he's currently at Cork College at the Elementary Pharmabiotics Center. And Jens came out of Gerald Tannick's lab at University of Otago, one of the top gut labs of the day back in the day. And that's Dan Peterson next to him. And Dan came out of Jeff Gordon's lab. Dan was an MD, PhD mucosal immunologist. And so we had myself, who was more of a genomics guy and becoming a quantitative genetics person, Bob Hudkins, who was a fermentation microbiologist, a true gut microbial ecologist, a young one, and Jens Walter, and then a mucosal immunologist all together. And we really had a lot of what we needed. The university invested in 454 sequencing technology, and we also had a germ-free mouse facility that Dan set up. All by about 2008, this was up and running here within our food science department. And I call those sort of the golden days, because during those days, a lot of the research we were doing was really low-hanging fruit. And I'll tell you just a couple of very quick stories of studies of host genetic factors, of host adaptation, that came out of the work during that period from this group and other collaborators. In 2015, we'll fast forward to 2015, the entire food science department at the University of Nebraska was moved out to a new campus, and this is Nebraska Innovation Campus that we're on. And this is a campus that's designed to promote public-private partnerships. And the university reasoned that the food science department had as good of a chance of anyone of trying to make that happen. And so they moved us out here and also challenged us, this gut function initiative, to become a little more than what we were, just a bunch of a cluster of researchers having a lot of fun and being able to get good funding for doing our work. And that led to the evolution of the Nebraska Food for Health Center. And I'll tell you a little more about that in a little bit, but that involved philanthropic investment, as well as university investments further into this group and into this area. So early on, we were looking as the gut function initiative at what factors really shape the species composition of the human gut microbiome. How does that work? And then, of course, what happens when things go awry? Most of us are well aware, right, this day and age, of the factors that we know about that are the common factors that influence composition of the gut microbiome. That is the maternal environment, whether the infant is breastfed or not, what the diet looks like after weaning and what the long-term diet looks like, individual genetics, how your individuality contributes to that composition, and then of course exposures to antimicrobials, antibiotics, other things that can impact the gut microbes. All of those things converge to shape the composition and the function of the gut microbiome. Early on, I started studying how your individual genetics influences gut microbiome composition. And I did this mostly in mouse models, not so much in humans that I have shown in the picture here, but you get the idea. And to make a long story short, we could do genetic analysis and we could actually demonstrate and identify loci in the mouse genome where genetic variation had a measurable impact on the human gut microbiome. And that was done largely through quantitative trait locus analysis or QTL analysis, and also through genome-wide association studies. The challenge with that was repeating that. And the reason was that the environmental signals, the environmental variation or impact on the microbiome was much greater than what the genetic effect was. And so you could pick up genetic effects of a population of animals grown in one environment. You could grow that very similar population of animals in another environment and get a completely different picture of how the genetic landscape was affecting the microbiome. And so that really was a challenge to continue working on that. We published a few papers on it that were very notable, and there's been some that have continued on looking in human populations and being able to study that, but it was quite difficult for us to sort of maintain that line. So we also recognized, of course, that there was a mountain of evidence showing that essentially you can trump any sort of genetic imprint or genetic effect, post-genetic effect on the human gut microbiome simply with a diet. By altering a dietary variable, you could completely alter what that genetic landscape or the effect of the genetic landscape looks like. And this was a growing area of strength within the UNL Gut Function Initiative. Several papers had been published for that group. In other words, the other guys in the group chose wisely. They chose to study dietary factors. I chose poorly. I chose to go after the genetic factors, which seemed more sexy at the time. So the bottom line was that we moved out to Innovation Campus and were challenged by administration and a couple of philanthropists to develop a vision for what the philanthropists termed at the time Agriculture 2.0. And this is going to involve diet. So let me give you a little background for where that vision came from. So if you look at the University of Nebraska or essentially any land grant institution, what you'll find is tremendous strengths in agricultural research. And these are just a few of the entities that we have here in Nebraska, but you can find many of these entities at University of Illinois, at, name your land grant institution. But powerful plant breeding and genetics programs, soil sciences, powerful ag engineering programs. Here we have the Drought Mitigation Center and Calmet, a land management center. All of these agricultural, what I call components that make up the agricultural engine, are focused exclusively on production, yield, and sustainability. Nutrition is not on that list. Now think about that. We have these amazing agricultural engines that can transform plants in ways through traditional breeding and genetics, can create robotic phenotyping strategies, can do remote sensing. All of this capacity is all focused on production, yield, and sustainability. Now, don't get me wrong, we have to do that, but can we make agriculture 2.0 where we extend beyond just production and yield and include human health and disease prevention? Can we include nutritional targets on this sort of vision? And so that became the vision that drove the development of the center. The center of the Nebraska Food for Health Center was established. Let's go back there. The Nebraska Food for Health Center was established via startup funding coming from philanthropy, as well as funding coming from the university for new positions and increased infrastructure. And altogether, it's about a $40 million endeavor. And the University of Nebraska Foundation is also committed to raising the balance of that to help the center grow and make it sustainable. Well, the center's vision is really focused on the gut microbiome as a part of this equation. And as most of you are very aware, the human gut microbiome is crucial for health and wellness. It's involved in development of the immune system early in life. It's involved in metabolism of things like dietary fibers, polyphenols, the dietary components that make it down into the colon. It produces energy for the colon, largely in the form of butyrate that colonocytes use as their primary energy source. And that production of butyrate comes from fermentation that's going on by the gut microbes. There's other beneficial metabolites that the organisms produce, some of them believed to be involved even in the gut-brain axis and communicating signals with the brain. It's involved in homeostasis of gut tissue functions and in maintaining that barrier. And it's also involved in resistance, colonization resistance to pathogens. Without that gut microbiome, you are very much susceptible to pathogens in a way that you normally are not. And abnormalities in composition and function of the microbiome over the last decade or so, or 2 decades almost now, have been shown to be associated with a wide range of diseases. There's just a few of them shown here, where disconfiguration of the microbiome is associated with this. And in some of these cases, we know that disconfiguration of the microbiome is actually a causal factor in the disease. And we know that because we can take a diseased microbiome from a diseased individual, you can transform or transfer that into a germ-free animal, such as a germ-free mouse. And over a short period of time, that animal will begin to take on characteristics of that disease. So we know, and like I said, in some cases, such as obesity and metabolic syndrome, that there's cleared causal roles for this disconfigured microbiome and disease. Well, if the microbiome is essentially eating our diet and is involved with wellness and disease prevention, then it provides a key link between agriculture and biomedicine. And if you appreciate the fact that these two areas are tremendously siloed, pardon the pun, and that normally agriculture and biomedical research are not working together. They're working independently of one another. And so the whole goal of the center is to make that happen. Well, that little cluster of 4 researchers that started back in 2007 has now grown into, this slide's a little bit dated. We've got 30 faculty members now that are part of the Nebraska Food for Health Center, 40-ish students at any one time and trainees, and 4 full-time staff. And we're supported by a wide range of philanthropic investments and gifts into the center, as well as individual grants that individual PIs are funded by. The Nebraska Food for Health Center is built upon a research platform that became part of the vision. And that platform connects breeding and genetics in plants, and to a little bit lesser degree, plant biochemistry and food processing, with early stage in vitro screening technology. And I'm going to take you through in detail a story of this and how we do this. But the idea is to do early stage screening in vitro, not in human clinical studies, because it's a lot more cost effective. It's much more rapid to do. And we can screen hundreds to thousands of different breeding lines of crops to identify lines that we're interested in. Once we've identified genetics or even chemistry that we're interested in that has a measurable impact in vitro on a gut microbiome, we then move that into gnotobiotic mouse models so we can humanize those mice with the same microbiomes that we're using in vitro to screen with. And that provides a very nice preclinical screen, as well as a model system for hypothesis testing. I won't have much time to talk about that today, but that is a really powerful model that we have in the center. Dr. Amanda Raymer-Tate runs that facility and has been an amazing colleague over the years. So out of that pops these novel microbiome active food molecules, as well as a better understanding and even new understanding of new and beneficial microbes. And at that point, things can go into human clinical studies. I'll also mention and take off my center director hat and put on another hat because I'm on a campus that promotes public-private partnerships. I'm also co-founder of Symbiotic Health. I hold equity interest in this company. Only to say that one of the roles of Symbiotic Health is to commercialize discovery that's coming out of this pipeline. And so we work through the university's intellectual property system to license technology coming out of this discovery platform and preclinical discovery platform. And Symbiotic Health pushes that farther down the clinical pathway. So we really go from if I go put my center director hat back on now, we go all the way from very early stage discovery through preclinical and early stage clinical evaluation into commercialization. And a lot of that happens right here just on this one campus. So I want to tell you a story now to give you an idea for how this pipeline works and start looking at how genetic diversity within a crop species actually affects microbiome active components. And many of you may not be used to thinking about genetic diversity within a crop species, but I've got pictures here that give you a really good idea for how much diversity really exists. Many of the contemporary species of crops that we grow and consume were domesticated 10,000 years ago. And through selective breeding, things changed over time, and they created tremendous ranges of cultivars with lots of unique different characteristics. I'll point to the dry beans down here on the bottom. This is a mixture of the dry beans that you might purchase at the grocery store. And what many of you probably didn't realize, and I didn't realize until I got into this, is that they're all originated from the same species known as Phaseolus vulgaris, or common bean. And you can see the phenotypic diversity in terms of size and color and shape of the seeds that were developed over time. And there's a breeding pattern that was used, a historical breeding pattern that was used that led to the origin of these seeds. And these seeds are categorized now into what's called market classes. So, for example, red beans are a market class, kidney beans are another market class, black beans are another market class. So all of those originated from the same species. So tremendous genetic diversity, as you might imagine. Maize thing up here, so looking at monocultures of maize that are clones growing out in the field. They're generally hybrids, but they're all very, very, very closely related. Traits include growing straight up and down and not growing bushy, and a lot of different seed traits that also in fact impact size, shape, color, and composition of the seed. Lastly, I'll point here to sorghum, which is the one we're going to talk about. And sorghum has beautiful genetic and phenotypic diversity that you can see. This is a diversity panel that was growing out in one of the plots here at the University of Nebraska a couple of years ago. And you can see the various ranges of color in the seeds, most of that controlled by polyphenols and tannin content, as well as tremendous diversity in height and even shape of the panicles. Just lots and lots of diversity in these seeds. So we started with sorghum, and we started with sorghum because it's well known for its diversity in fiber and polyphenol content. There's a number of bioactives that have been identified with, now we don't understand the microbiome component of that, but we certainly know a good bit about how some of those bioactives function on human cells. It's consumed daily by certain global populations, and it's a crop that we can actually readily cultivate here in Nebraska, especially in Western Nebraska. So I'm going to talk to you now about using the human gut microbiome as a health trait for food crops. And the idea here is to screen genetic diversity. And just like you would map a trait such as seed coat color and link it with variation in the genome on the different segments of the chromosome and link it to variation in the trait of seed color, we're going to use the human gut microbiome as a proxy for that. And I'm going to show you how we do that. And ultimately, through that study, we were able to go all the way back to causal variants and identify the exact mutation that was causing the changes in the microbiome, and therefore understand the pathway through which this is working. So how can you begin to do this? Well, first of all, it's impossible to screen hundreds to thousands of lines of genetically diverse crop against humans, because I can't feed each human each different line. It's simply an impossible experiment to do. But what I can do is I can take a human stool microbiome and I can aliquot it across lots of different wells. There are lots of different test tubes essentially. And I can then screen how that microbiome in vitro ferments each of these different seeds. So I can get an individual look at how a microbiome will ferment or utilize the components that are present in these different seeds. And we took this system, this in vitro fermentation, and we miniaturized it and we automated it. And it's referred to now as automated in vitro microbiome screening, or AIMS. And the way AIMS works is we take the grains. The grains are milled, and they're milled individually by line. And the milled grains are then introduced into 96 well plates at that point. They're taken through acid hydrolysis and enzymatic digestion to make it look like digestion that's going on in the upper GI tract. That includes addition of pancreatin, glucoamylases to degrade both protein, lipid, and complex carbohydrate content. We then take the digested material through dialysis, and we use 1,000 molecular weight cutoff membranes for that dialysis step. And what that does is it removes the monomers from the reaction. It removes the glucose. It removes the free amino acids that have been part of the digestion process. And the reason for that is those are normally absorbed in the upper GI tract, and we only want what enters into the colon. So what remains after dialysis then is introduced into the stool microbiomes for the fermentation reactions. So that's the process. If you want to see, we've got video of some of that process, and I'll leave this up here. You can take a picture of this. I wouldn't suggest going there now because it'll distract you a little bit. But if you take a video, this QR code will take you to our website where there's some video of this system that you can watch. And I will make a public service announcement that when we made this video, I did put some gnarly loud rock and roll music behind it. So if you don't like that, turn your volume down. If you do like it, turn it up because you might enjoy it. Okay. So the thing about this AIMS approach is we can scale it multidimensionally. We can either screen a very small number of human microbiomes against a very large population of seed or plants. And there we're looking at plant population level factors. So we're doing GWAS mapping, looking at genetic bioenvironmental factors and how that impacts the microbiome. The other alternative is once you find lines that are of interest now, you can focus on a small number of lines, and then you can screen those across a larger population of human subjects. So you can scale this AIMS process multiple ways, and it's a very, very powerful system to have in place. So I'll tell you about the first proof of concept study that we did in sorghum. This was published in Nature Communications towards the end of last year. And it really stood out. The Nature Communications editor loved it. It was one of the featured articles from the editor's features. He loved it because of the novelty behind it. And so this is a study that was done in collaboration with James Schnabel, who's one of my favorite plant geneticists here on campus. I collaborate very closely with Scott Sattler for the ARS, who's also a sorghum geneticist, Devin Rose, who's within our food science department here, and Devin is a food chemist and also a microbiome person. Came from Bruce Hamaker's lab. Very, very wonderful collaborator. Qinan Yang was the graduate student that led this, and two other graduate students, Nate Korth and Mallory Van Hout, also contributed to this as well as their own projects they were working on. So a little bit about how this works. So the first study that we did was a set of recombinant inbred lines, and I'll explain that to you here in just a second, that originated from a cross between two different parents, BTX 623 and IS 3620C. And these two parental lines of sorghum had a lot of very unique characteristics that differentiated them from one another. This population was developed several years ago and has been used for multiple studies. So the way the recombinant inbred lines work is that the cross is made between the two parents, and individual F1 progeny then are begun to be inbred after the F2 generation. And you go through inbreeding for roughly 6 generations, and you have about 95% homozygosity across the chromosome. And so each of these recombinant inbred lines now is a random assortment, a random mixture, a combination of chromosomes from one parent and the other, which I've colored here. And each one is different, and each rill is its own clone, meaning that its progeny will be identical as long as I don't cross them with anyone else. So this is a powerful population in which you can study assortment of different traits. And the way, sorry about that. The way we approached studying this was, as again, to use the AIM system, was to take seed from each recombinant inbred line, mill it up into powder, as I said, put it in the 96 well plates, run it through this in vitro digestion reaction, dispense the stool sample into the wells after that's done. And then we use DNA sequencing, 16S our DNA sequencing, to monitor composition and quantify composition of the microbiome after fermentation. And as you might imagine, the genetic variation in some of the seed lines actually causes tremendous variation in the fermentation patterns, and that shows up in the unique combination of organisms and their relative abundances. Those microbe abundances then become our traits, and we do genetic association analysis. We have genotypes across the parental lines. In fact, both parental lines are fully sequenced. We have genotypes across all of the recombinant inbred lines, and it's a simple statistical association between variation in the microbial abundances of individual taxa or groups of taxa and genetic variation that's happening in the chromosome of the recombinant inbred line. So that's the strategy that we use. Just real quickly, a little bit about the two parents. The IS3620C comes from a Guinea race, which is a land race that was domesticated a long time, or well, thousands of years ago. And BTX623, which is actually a blend between two other land races, Kafiran and a Caudatum blend. And so this would almost be, the equivalent of looking at two very different serovars of Salmonella. You can kind of think of it that way, but on a grander scale in terms of the genome size and the amount of diversity between those two parents. As I mentioned, this is a well-studied population. Lots of different traits have been mapped in this population, which is why we chose to focus on it initially. As you'll see, we got very, very lucky that we chose this population. So here's the two parents, 623 and IS3620. The first thing we did was to run the two parents across microbiomes, just the parents, across the microbiomes of 12 different essentially randomly selected human subjects that had not been on probiotics, had not been on probiotics, and otherwise had a healthy GI microbiome. And the heat map here shows you the log fold difference between one parent versus the other. And if it's extremely red or extremely blue, then that microbiome is really differentiating those two parents' lines from one another. And so we chose one of the subjects here, microbiomes, based on essentially the average reaction, the average differences that we were seeing. And we then use that single microbiome to screen roughly 300 different recombinant inbred lines from that cross. The lines were the lines from the cross, the recombinant inbred lines were grown in the greenhouse to reduce any impact of environmental factors. As I said, we chose one human microbiome to screen with initially. That was subject 765. We always do these in triplicate. So there's 3 line by microbiome replications. So you can see the scales relatively quickly. 16S rDNA sequencing is used for phenotyping the microbiome across the V4 region. We also, in each of the reactions, measured metabolites. So short chain fatty acids were measured in each of the reactions. And the abundances of individual taxa, metabolites, and groups of taxa become our traits now for the QTL analysis. And for the QTL analysis, we use 616 informative markers for each line that was part of the mapping. And I will say, because many of the microbiome traits are non-parametrically distributed, we use non-parametric QTL mapping algorithm that's in our QTL. So this is what this data looks like when you put it all together. And this was a way we came up with visualizing the data that sort of helps us. So each ring here is a different set of microbial phenotypes. The first three inner rings are metabolites, short chain fatty acid profile. So butyrate, valerate, for example, those are the first three rings. The next three outer, or the next bunch of outer rings here are different families of microbes in the microbiome. And the segments, 1, 2, 3, those are the different chromosomes of sorghum. And the scale on the y-axis here, these are essentially little graphs that we put on top of one another. The scale there is the significance of the association. It's a logarithm of difference from the QTL mapping program. So anywhere you see peaks starting to show up here indicates there's a significant association of variation on the chromosome in those recombinant inbred lines and variation in our microbiome traits. And what you'll start to notice pretty quickly here is that we had regions on chromosome 4 in particular, another one on chromosome 2 right here, where multiple microbial traits are stacking up on one another. So variation in the same region of the sorghum genome is influencing the abundances of multiple organisms within those fermentation reactions. And we refer to these regions as multiple effect loci because they're overlapping QTLs, they're overlapping significant associations for multiple microbial taxa. And to make a long story short, what we've learned is when you're using small numbers of microbiomes to do this screening with, focusing on these MELs, these regions of multiple effect loci, lead you ultimately to high impact genetic variation in the host, in this case sorghum, that has a whopping effect on the microbiome. And when you subsequently screen that effective variation on multiple microbiomes, you see a very similar signature. And I'll show you an example of that here in just a minute. So that's the concept of a MEL. That's how we're doing our mapping strategy. What you might not have necessarily noticed, but I will point out here, is that on the different chromosomes, if we look at our metabolite profiles underneath here, we have regions where metabolites are stacking up, and they're also stacking up with other microbes. There's one there on chromosome 2, another one here on chromosome 5, where we have traits coming from the metabolite profiles and microbes that are largely responsible for generating those. And here's the actual traces. If we zoom in and look at the traces, you can see those peaks are right on top of one another. So those are instances where we think the variation in the sorghum genome is affecting the microbe, and the microbe is influencing the amount of that particular metabolite that's showing up. Okay, so now the fun part of the story. So this is where we got really lucky, because it turns out my geneticist collaborator, James Schnabel, happened to notice that the positions of the peaks that we had, the MELs that we had on chromosome 2 and chromosome 4, were right over the regulatory genes known as TAN1 and TAN2 that control synthesis of condensed tannins that are made by sorghum. The peaks are right on top of them. And so we went ahead and looked at color. So we mapped the first principal component of RGB color that we measured on each of the individual seeds from each individual line. And there we get nice peaks on chromosome 2 and chromosome 4. We measured actual tannin content and again got nice peaks on chromosome 2 and chromosome 4. And then here's relative abundance of Faecalibacterium from the microbiome that we're screening with. Again, nice peaks on 2 and the whopping peak on chromosome 4. And then if we use qPCR and measure absolute abundance of Faecalibacterium and use that a trait, again, you can see this whopping overlap, this peak on chromosome 4. And these all align beautifully with one another, suggesting that indeed this could be variation in the tannin regulatory genes that's controlling tannin biosynthesis, and tannin is actually what's contributing to our microbial phenotype. So it turns out that TAN1 and TAN2 are both regulators of the condensed tannin or the proanthocyanidin biosynthetic pathway that leads to the production of condensed tannins. Both TAN1 and TAN2 must be functional for the pathway to be active. A mutation in either one of those genes will block the pathway. It's known as duplicate recessive epistasis. So, if you might recall, our parents of the population were both white. They both produce cream-colored seeds. And the reason they do so is because the IS3620C parent has a mutation in TAN2 that is a null mutation that knocks that gene out. It's a 95 base deletion. The BTX623 parent has a mutation in the TAN1 gene that's also a null mutation that knocks that gene out. So both parents are unable to synthesize tannins because they have a mutation in one or the other regulator. But what that means is that they have wild type intact copies of the other regulator. So IS3620C has an intact copy of TAN1 on chromosome 4. It has a mutant copy of TAN2 on chromosome 2. In contrast, 623 has a mutant copy of TAN1 and has a wild type copy of TAN2. And what that means is some of the progeny are inheriting wild type copies of both genes, functional copies from each parent, leading to about a quarter of our progeny that are actually producing tannins. And what fascinated me was that nobody had actually noticed this in this population before, and nobody had reported this particular trait in this population. So if we measure tannins now, and we now group the recombinant inbred lines based on their genotype, so the three categories of tannin negatives that have a mutation in TAN1, have a mutation in TAN2, or have a mutation in both genes, all produce cream. And then the quarter of the lines that produce wild type TAN1, TAN2. If you measure tannin content, you see that it's very high only in the TAN1, TAN2 genotypes. Same thing with seed color, same thing with Faecalibacterium, Roseburia, and a number of other organisms that also map to that MEL. So this suggests that indeed it is the tannin content that's driving this strong microbiome phenotype. Now I'm gonna pick it up just a little bit so that we don't run out of time, but I want to tell you that one of the other luxuries we had on campus was that Scott Sattler, another collaborator of ours, had generated a set of isogenic lines that differ in sorghum, that differ only by what's present at the TAN1 locus. So he essentially introduced the TAN1B mutation into a wild type tannin background, and we had lines that were developed through back crossing that are genetically identical at every other locus except the TAN1B. So when we now take pools of recombinant inbred lines with the different TAN1, TAN2 genotypes, so we created 4 pools, and then we compare those to the near isogenic lines, what you get is a very strikingly similar signature on the microbiome, and this is now screened across 12 different human subjects. And so the red versus blue here represents the mean fold change between the tannin positive and tannin negative. Here you're looking at the recombinant inbred lines. Here you're looking at the near isogenic lines, or the isogenic lines. And you can see that the patterns here are very, very similar. We've sorted them by blue on top and reddish on the bottom. And perhaps the easiest way to see this is to look at the correlation. And so here you're looking at the correlation at the family level between a log fold change in the nils and a log fold change in the recombinant inbred lines. And the correlation is really quite staggering. So again, suggesting that it is that variation, that knockout in that TAN1B gene that's driving our microbiome phenotypes. Well, one last thing to put a nail in the coffin. We did molecular complementation where we took purified tannins and added them back to the tannin negative seed powders to ask if we could actually restore the phenotype with pure tannins. And the answer to that, and we used both tannins that was extracted from sorghum as well as condensed tannins that were coming from quebarco trees or hardwood trees. And again, if you just focus on the correlation plot here, if you look at the log twofold change in the non-tannin rills in which we'd introduced the quebarco tannin versus the log fold change that we saw in the rills by themselves, you see the correlation again is outstanding, very highly significant. And you can see it in the pictures or in the heat maps that we have shown up here as well. So again, very, very powerful evidence now that it is the tannins that's driving this particular phenotype. And one last story to really drive the nail in the coffin was we took the tannins, and in this case, we just focused on the quebarco tannins. We introduced those into either microbiomes or a pure culture of Faecalibacterium prosnitzii, which was one of the organisms that responds. And we monitored we monitored growth of Faecalibacterium using qPCR. And so the baselines for where the experiment starts, the no tannin control, and then green is with the tannin. And you can see that in these different microbiomes coming from these different subjects, from all but one subject, we saw tannin stimulate growth of Faecalibacterium. And then the best evidence, of course, is this is a pure culture of Faecalibacterium. And you can see its growth is also stimulated when we put the quebarco tannin in. And lastly, we went to a traditional microbiological assay that I'm sure many of you will recognize, in which we created solid media with an overlay of tannin-containing solid media. And then you'll note that where we have growth of Faecalibacterium, you see these zones of clearing where it's degrading that tannin from its colored form into a colorless product. So again, good evidence that this organism is actually degrading the tannins. Well, quickly, I want to talk just a little bit about how this actually might relate to the world, because we don't eat a lot of sorghum here in the United States, but I want to show you that discoveries like this can actually be very, very significant in terms of human health. So Faecalibacterium, Roseberia, and other organisms that were influenced by the genetics, that were influenced by the tannin variation in that study I just showed you, these are all considered beneficial microbes. Most of them are associated with reduced susceptibility to inflammatory bowel disease or metabolic disease. Faecalibacterium in particular is one that is typically depleted in patients with inflammatory bowel diseases. So it's believed to be a part of a system that is talking to the immune system and reducing the chance of inflammation, reducing the likelihood that person might develop inflammatory bowel disease. So these are beneficial microbes that are being stimulated by the presence of tannins. And if no tannins there, of course, they're not stimulated. And so how would this relate to the real world? Where it turns out that in sub-Saharan Africa, sorghum is a major staple of the diet. And depending upon where you live in sub-Saharan Africa, dictates the type of sorghum that you are growing. And this is a fascinating paper that was put out by Jianran Li and Jingman Yu just a few years ago in Nature Plants. And what they noticed in this paper was that birds like sorghum seed also, but they don't just like any line. So they were growing diversity plots of sorghum out in the field. And what they noticed was that the bird damage was very much non-random. And to make a long story short, the birds went after the panicles that were the highest, so the tallest plants, and they went after the panicles that were not producing tannins. They don't like tannins because they have a bitter, astringent flavor to them. And so they actually mapped bird damage in the population they were studying, and lo and behold, the bird damage actually mapped a variation in TAN1 and TAN2. Same alleles in this population that are controlling tannin biosynthesis that we're controlling them in our recombinant inbred lines. Well, in Africa, there's a bird called Quelea. That's the most hated bird in Africa. And this is a bird that loves sorghum seed and literally swarms like insects and can destroy entire sorghum crops. So where they're growing sorghum and consuming it locally, this bird is a real pest, and it's a real problem. And in areas where that bird population is high, which is shown on this graph here from that paper, or this map, I should say, here from that paper, they tend to grow high tannin producing sorghum. So if you look at the graphs right here, this is the relative proportion of individual plots growing high tannin sorghum versus low tannin sorghum. In areas where that bird population is high, they favor growing high tannin sorghum because it's less susceptible to the bird damage. In areas where the bird population is low, they tend to grow non-tannin or low tannin sorghum. And the reason is because the flavor is much better. And it turns out that it's even more fascinating than that, because, again, to make a long story short, if you look at the taste receptor allele that's responsible for tasting bitter flavors and differentiating bitter flavors, the human populations that are in areas where they're growing high tannin sorghum tend to have a certain allele of a bitter taste receptor where they don't taste the bitter flavor. And that's how they're able to consume that on a daily basis. So it's this fascinating story of this co-adaptation between agriculture, between animals and pest populations that influences the agriculture and human populations as well, all co-evolving and co-adapting in these same environments. And I'll just point out that these same tannin alleles, the same alleles that knock out tannin production, are found in the plant populations that are growing there, are the same ones that were present in the recombinant inbred lines that we were looking at. So we have these complex interactions between the tannin regulators that are controlling tannin biosynthesis, as well as other things, including fatty acids and some volatile fatty acids. The tannin producing plants the birds tend to avoid. The tannin non-producing plants that have mutations that block one of these tannin regulators, those the birds like, and so do the humans like as well. As I mentioned, the human populations also vary in their taste receptor alleles, but the important point to remember is that humans in this region here in sub-Saharan Africa are consuming high tannin sorghum on a daily basis, whereas the populations over here of humans are consuming low tannin sorghum. Now that we know there's this strong microbial phenotype in the gut microbiome, where the tannins are stimulating growth of beneficial microbes, inhibiting growth of less beneficial microbes, and now you have human populations that are stratified based on whether they're consuming the high tannin or no tannin sorghum, it leads you to wonder, is there a human health outcome? I don't know the answer to that yet, but it sure makes me wonder about that question. So in summary, I think you can see from that first study where we've gone all the way from discovery through being able to define a phenotype, link it to a genotype, link it to variation in a specific gene with causal variation, show that causal variant truly does drive the phenotype, validate it in multiple systems, and now extrapolate that to importance that we might have in a food system and important potential effects on human health. I think you can see now how that discovery pipeline for the Food for Health Center was really set up to allow us to pull these things out of the populations. We've done mapping in another sorghum population now, and I just wanted to show you real quickly that in this population, we've mapped not only microbiome traits, but also 200 other agronomic and biochemical and seed traits. And so we're looking at the overlaps now between the microbiome characteristics and the seed characteristics. Powerful, powerful data set. We recovered a lot of the MELs that we picked up in the first study, and not surprisingly found several more. Why that did that, but let's go back one more. And lastly, that we've played this game now, not just in sorghum, but we've played this game in maize. We've played this game with other traits. We've looked at seed protein traits. We've also looked at starch traits. And in each case, we've shown whopping effects on the human gut microbiome. So I don't have time to describe all those to you, but those were all published within the last year. So hopefully I think you've seen from the story here how one can literally go from a vision of putting a center together, a vision that could drive the science of the center, and then putting that proof of concept study out there to illustrate how this actually works. Hopefully you can appreciate that occasionally you get really lucky and you either have resources on campus that somebody created for a completely different purpose that actually serve a purpose for you. And you get really lucky in having powerful collaborators with you on campus to work with. And I'll just end by thanking my amazing collaborators that were part of this study, James Schnabel, Devin Rose, and Scott Sattler. And then the 3 graduate students that were part of the study that I told you the most about. Jinan Yang was the lead on that particular study. And then a special thanks to our donors and our funding entities that have supported this project and support the Nebraska Food for Health Center. Thank you.
Thank you, Andy. Let's open for questions. I see some hands up. Hammerstone, I don't know if I pronounced your name correctly, please.
You did. Thank you. Thank you very much. Dr. Benson, I have to tell you, I was sitting on the edge of my seat for the entire presentation. I actually had sorghum for lunch today.
Oh.
I have a small snack food company with my business partner, and it's based on sorghum as our hero grain. And I've done research in the past on proanthocyanidin, so this was really exciting for me. My question is, have you looked at the effect of processing and how that influences the outcome of the changes in populations?
We're just starting to do that work. I've got a collaboration with the ARS unit down at Kansas State. That's a great sorghum unit down there. And Dmitry Smolensky is the individual. So we're just starting that because that's an obvious next question for any of these traits, right? Is how does processing affect it? So stay tuned.
One quick follow-up. Are you working with any commercial breeders for these lines or no?
Not at this point, but I'm certainly in the process of finding out who commercial breeders are, because as you might imagine, you scale very quickly from needing 10 grams of seed to needing hundreds of kilograms of seed to do a human feeding study with. So
okay.
So we definitely are looking at that.
All right, thank you.
Yep.
And I just have a couple of quick comments. As usual, listen to your seminar or research talk is so always overwhelmingly informative and eye-opening experience. I'm curious, what's the overall turnaround time for your study? I know the plant side would be the most time-consuming, I would suppose. The microbiome, the kind of metabolism side would be relatively quick. And what's your criteria for selecting human subjects? You didn't mention, you said randomly selected a number of human subjects with healthy
Yeah, that's a good question. So you're right in that the turnaround time is often the longest for production of seed. But the great thing is we only need small quantities, so 10 grams of each line. So most of the time, if I go and talk to the plant geneticists or I go to the germplasm resource centers, I can get that much seed right away. They've got plenty of seed sitting around. So oftentimes that's not necessarily the limiting factor unless I have to grow it up, but I might have to grow it just one generation. The actual Ames part of it, the microbiome part of it itself, even with all the automation, it's still time consuming. So we typically run about 2 months to run a set of 300 lines against a couple of different microbiomes. That gives you a little perspective of what that takes. That's just to generate the data. And so then there's another month of iterative data analysis that goes on with that. And then the second question, with respect to choosing microbiomes, essentially we're trying to stratify right now based upon dietary pattern, based upon race, ethnicity, if we can. So we're collecting all that information when we're recruiting subjects. And then again, of course, no recent history of probiotic use, no recent history of antibiotic exposure. But that's the only criteria right now. One of the challenges that we have faced, and it's a legitimate critique, is that right now we're using healthy human microbiomes. What about disease microbiomes? And so one of the powerful collaborations I have is with the Posture Inflammatory Bowel Disease Center, which is at our medical center here. And we're just starting on that process of looking at disease microbiomes also.
Yeah, that's the thing I try to follow up.
Yeah,
Exactly, to study the diseased people
right.
with chronic problems such as obesity, and that data can be part of the basis for like a personalized food choices and things like that.
Exactly.
That's invaluable. Yes. Jiang Kong.
Hi, Andy. Finally to talk. A question about the recommendation. Do you recommend a healthy individual to take probiotics?
Would I recommend that? Are you saying?
Oh yeah, would you recommend that? I mean, it seems like there are so many products out there in the stores, and people, they heard about how good to your health.
Right.
Even though they are healthy, they're normal, but they're taking those probiotics.
Yeah, that's a fascinating question. If you look at a lot of the probiotics that are currently on the market, what I will tell you is that I'm with you and that the efficacy might be questionable. I do think there are individuals where you do see an effect. I think they're probably fewer and farther between. And I think that's why a lot of the clinical studies look the way they do, that you have a few individuals that experience significant effects from it, and then most individuals who don't. And if you happen to have enough of those individuals in your study, you achieve significance. I do think there are some benefits that you can certainly see and think about the mechanisms through which those benefits would happen. But what I think is more exciting is the next generation of probiotics that's coming. And now I have my company hat on while I'm telling you this, because the second generation of probiotics was chosen on very specific criteria, not whether I could produce it in a massive fermenter or not, although that is one criteria. You still have to be able to do that. So the second generation, so to speak, of probiotics, a good example that's not from our company is the product that's on the market by Pendulum. That's got Akkermansia in it. And Akkermansia, there's good clinical data behind that, being very supportive of individuals in reducing insulin and insulin resistance in patients with diabetes. And so there's an organism with very good clinical evidence behind it for efficacy. I think there's a lot of excitement around that product that's out there on the market right now, and its ability to reduce symptoms of diabetes. And so I think this is the next generation that you're going to see. They're probiotics that have been chosen for very specific reasons, and even symbiotic combinations. I come back to the tannin story. Faecalibacterium is the one organism that's usually depleted in anybody with inflammatory bowel disease. And so now you have at least one thing in the diet, i.e. tannins, that stimulates this organism. So you can think about creating some really novel symbiotic combinations, and maybe that would allow you to reintroduce that organism into a disease microbiome. I mean, we'll see, there's a lot of work to be done there. So the symbiotics are going to be a fascinating sort of new avenue to see if we can improve efficacy and have products on the market that truly are science-driven and efficacious.
Thank you, Andy.
Yep.
And Bezar, you have questions, please?
Andy, great to see you again. It was a great presentation. Fantastic science. I think you presented really well. I have two quick questions. In the beginning, you started with 454 sequencing, and can I ask you do do you have your own sequencing center right now in the college?
Yeah, so we have our own MySeq that's the centers. It happens to be in my lab, but the center uses it. And the way we operate that is we have essentially one person from each lab that is the sequencer, that is approved on it, and they do the runs on there. The labs are all responsible for their own bioinformatics. And the center has 3 biostatisticians, faculty members that are part of our center. And then we have 2 bioinformatics faculty that are part of our center, actually both in the food science department here. So we've created a culture here where the students learn the bioinformatics essentially on their own, but by networking and by taking courses. And so pretty much everything gets done within the center, and we really don't have any core facilities that we utilize. We do have a good bioinformatics core here on campus, but we essentially just do all of our own stuff.
My second question is about how do you keep your donors interested in your work throughout the years? I think it's a big challenge, as you mentioned.
Yeah, that's a trade secret. I don't know if I can share that, but no, I tell you, one of the things we do is we do a lot of show and tell. So like last year, one of our donors that came in, we brought all of these different sweet corns and popcorns out of the field that one of our breeders was developing. And James Schnabel spent time telling, and of course they had all the different colored kernels and everything in there. And so James spent good time with the donors telling them how that happens, why you end up with these different colored kernels on it. And they're fascinated by that. The other thing that we do is I make, usually on about a quarterly basis, we make and share update videos with our donors. And like the last one I did featured some of our fellows that are grad students that are Food for Health fellows. And the fellows talked about their work and, the research that they're doing. And so it's communication with them. It really is. And I'm very fortunate. I have a project manager who worked in that realm in philanthropy. So she's very much able to take me down off of the scientific pedestal and talk at the donor level. And so we've learned how to do that over the years too. And I think I'm still learning how to do that, but it's an important part of the process.
Thank you.
Any additional questions? Andy, I see your last slide. You have a lot of funding. Well, I don't know how much funding you get, but you have a lot of entities to donate money to your research, and most of these are research foundations.
Yes.
You mentioned about the PPP, the public-private partnership. I wonder if there is any companies like food companies, biotech companies, pharmaceutical companies who would fund you or kind of join as a member and get benefits out of your research?
Starting to. So that started after the Nature paper came out. We got a lot of press out of that paper. There was companies that contacted us very quickly after that. And just a quick little aside story, the 96 well dialysis plates that we use that have the dialysis membranes in the bottom, the manufacturer quit making those right after we published that paper because we were the only customer. But since they found out there was so much interest in those, they've started to remanufacture those. So yes, we're definitely trying to grow that realm.
Sounds great. And probably for those, as Jian Hong mentioned, there are so many brands and companies doing these commercial probiotic dietary supplements. Actually, I bought quite a few for my dad. And he seems like he likes it, and he thinks he has those has a kind of beneficial effect on him.
Yep.
So I wonder if those companies would like to do some validation study with you.
Oh yeah.
Kind of prove, have scientific evidence that my product actually works.
Well,
So,
you can actually use this strategy for very early stage strain discovery, right? And it's a good strategy for identifying combinations of strains and then I won't necessarily limit it to prebiotics, but let's say prebiotics, polyphenols, other types of molecules that could be used to pair with these things that will stimulate their growth. So there's a lot of interest in doing this right now.
Yeah. Well, I think we're approaching 2. Thank you so much, Andy, for giving us such an informative seminar. I'm gonna take a look back to your slides, and I can digest some more information. I probably grab like a quarter of the information we presented. It's too fast, too much data. So anyway, thank you so much for joining us and give us a seminar. And I look forward to follow up with you for some collaborations.
Good. It's great to see everyone. And again, and I know it's been a while. I miss the old days, the whole genome sequencing meetings there's at iFish. I remember those well. Those were good. So
Okay.
We got one coming up at the end of May.
What's that?
We have one coming up end of May.
Okay, so they're still going on.
Yes, this will be the first one back in person, right, Bazad?
Yes, that's right.
Finally.
Yeah, May 31st and June 1.
Yeah, but you to be
I
our
hope you
speaker
can
next
come,
month.
and it's a great, I think, presence. Good.
Well, I've got to get back there one of these years. I've still dabbled a little bit in population genetics. I had one postdoc that wanted to come train with me and insisted on coming and training with me, and I said okay. And so we published about 3 or 4 papers together, putting a very robust system together for doing really high throughput population genomics. So I still dabble in it a little bit, but it's not my main thrust anymore. Good to see everyone. Thank you.
Thank you, Andy.
Thanks.
Thank you.
Thank you,
Thank you everyone for
joining us.
attending.
Okay, see you later.
Bye.
Bye.